Okay, in continuation with the previous class, today we'll try to complete erythropoiesis, its regulating factor, and how these factors govern and control erythropoiesis. Okay, क्या हुआ तभी ठीक नहीं? Okay, so when we're talking about RBC structure, okay, in previous class about its biconcave nature, what were the merits related to it, okay, so that was a normal shape of RBC, okay. So these are certain terms which will come under regular frequency. We frequently will come in contact with these terms, so you should have certain idea about it, okay. But along with this, when we are talking of counts, okay, there are variation in counts, and these variation may result due to physiological reasons, maybe due to pathological reasons, okay. When we are talking of physiological variations, the causes may be diurnal, more in the evening than morning. So this is just a physiological variation. Exercise, it increases RBC count. Plus indicates it increases the count. High altitude, definitely it increases the RBC count. Temperature, hypoxia is a pathological condition, okay, not a physiological. It at times it may appear physiological. Okay, hypoxia, adrenaline excitement, meals, age, and gender. These are the certain conditions in which the variation in RBC count is seen. Now, what do you mean by variation in shape? When there is variation in the shape of RBC, it is known as poikilocytosis. Now, what are the different variations that can be seen? Crenation or shrinkage of RBC, they are known as burst cells. Okay. Spherocyte, we've seen spherocytic anemia, in which the cell shape appears spherocyte. Ellipsocyte, sickle cell, poikilocyte. So these are the terms. जब अपन एनीमिया का क्लासिफिकेशन करेंगे ना, तब अपन इन टर्म्स को यूज करेंगे फ्रीक्वेंटली. तो ये एक पिक्चर, ये these terms will remain as such. You cannot do more. इसको अपन हिंदी में कुछ ट्रांसलेट करके नहीं दे सकते. ये जो टर्मिनोलॉजी है ये यही रहेगी. क्लियर? और फ्रीक्वेंटली जब अपन एनीमिया इसका अप्लाइड एस्पेक्ट करेंगे तो ये टर्म्स हमारे अंदर क्लियर होना चाहिए. ओके, वेरिएशन इन साइज. Previous was variation in shape. It is variation in size and isocytosis. When there is variation in size, the size may be normal. The cell will become normal size. Okay. When the size is bigger, macro size. When it is smaller, it is micro size. So in particular anemia, if the size of the RBC is small, microcytic type of anemia. If the size of RBC is more, it becomes macrocytic type of anemia. So these terms will come when we go for applied aspect. So this concept should be clear: variation in size and variation in shape. Okay. This is the metabolism of RBCs. Glucose taken up by the facilitated diffusion. So it is only the fuel utilized by RBCs. Okay. These are the different pathways by which the metabolism or the energy requirement is done, and these are the various enzymes which are present. This is, we have started with hemopoiesis. It may be erythropoiesis, leukopoiesis, or thrombopoiesis. Okay. In embryonic life, we have seen extravascular and intravascular theory. Now, this is the nomenclature when we are talking of erythropoiesis. So, the different stages, they are called by different schools, if it is the British school or the American school. Depending on the school, okay, the different terms are there, though the stages will remain the same. जो सेल के अंदर चेंजेस आ रहे हैं वो सेम रहेंगे, but their nomenclature is different. Okay, so whichever book you are dealing or whichever thing you are going for, एक ही पे follow करना. ये नहीं कि आधे ब्रिटिश के ले लिया, आधे अमेरिकन के ले लिए. Either go for British or either go for American. Okay, whichever the series is there in your book, but don't combine the thing. Yeah. So when we are talking of British school, it is hemocytoblast, endothelial cells. That is the different. Okay. But more or less, it is proerythroblast, then Here it is megaloblast, early, intermediate, late, same terminology over here. Okay. Now, stages and maturation of erythropoiesis. So when the story starts in hemopoietic stem cells, okay, when we are studying hemopoietic, the bone marrow, okay, out of 1,000 cells, only 2 or 4 hemopoietic stem cells are seen. These cells cannot be seen by simple microscope. Okay, advanced technology is required for their identification seen under microscope. These hemopoietic stem cells, when they are under the influence of certain hormones, proteins, okay, now what is their role? They will identify and they will be committed for a particular series. Here we are concerned with the erythroid series. Okay, so certain growth inducers, differentiators, these particular proteins will result and will affect the hemopoietic stem cells to multiply mitotically and result into the formation of colony forming unit responsible for erythroid series. Here both the division and maturation, okay, is taking place. Now what type of maturation, division? Okay, hemopoietic stem cell or the first stage proerythroblast. It is the first cell which is identifiable as the first line of erythroid series. Okay, we cannot differentiate the pluripotent hemopoietic cell as the first line of erythroid series because it has to commit for a particular series, whether it is a lymphocyte, whether it is a platelet. So in MCQ when it comes, which is the first line to be identified as the cell for erythropoiesis, okay, or the erythroid series is the proerythroblast. Okay, blast is a nucleated cell which is committed for further mitotic division to take place. Okay, this under the influence of certain proteins, okay, they will differentiate, grow and result into the erythroblast. This erythroblast is written as basophilic erythroblast. Now why we are calling it as basophilic? Due to dye. But why the dye, particular dye is being taken by the cell? Okay, staining, okay, but the content has to be there. Okay, बेसो इसलिए क्योंकि ये क्या होता है, सेंट्रल ब्लास्ट है तो न्यूक्लियस तो डेफिनेटली होगा. इसके न्यूक्लियस के अंदर क्रोमोसोम्स जो होता है ओपन फॉर्म में होते हैं, यूकैरियोटिक न्यूक्लियस हो गया. पर इस सेल को क्या बनाना है, मैच्योर होके हीमोग्लोबिन. तो मेन कंटेंट ऑफ हीमोग्लोबिन इज globin, heme. Heme requires iron, protoporphyrin molecules. Globin requires proteins. For the synthesis of proteins, what we require is the ribosome. Okay. So for synthesis of ribosomes, we need the idea of mRNA. So mRNA and ribosomes concentration is more in the cytoplasm. RNA is a acid, so it will take the basic stain. So the content of this mRNA and ribosome in the cytoplasm of the proerythroblast will result into the basophilic nature of this cell. That is why it is known as basophil erythroblast. Okay. After this, what will happen? Continuous mitotic division is taking place. Till what stage? Till the intermediate stage or the polychromatophil erythroblast, the mitotic division continues. Okay. After intermediate stage or the polychromatophilic erythroblast stage, the mitotic division ceases and now the the maturation takes place. Okay. Maturation शुरू से हो रहा है पर यहाँ पे mitotic division stop हो चुका है. Now further growth will take place, maturation changes will take place, okay, but no more mitotic division will take place after intermediate or polychromatophilic erythroblast. Okay. Now why we are calling it polychromatophilic? Because the ribosome से mRNA जो cytoplasm में present था उसने अपना function start कर दिया. Now it has started forming hemoglobin. So hemoglobin is eosinophilic. It will love the acidic stain. It will give reddish appearance after staining. So the cell will appear bluish and reddish. तो ये सेल क्या कर रहा है, दो स्टेन को लाइक कर रहा है, सो इट इज पॉलीक्रोमेटोफिलिक एरिथ्रोब्लास्ट. ओके, अभी अपन केमिकल नेचर की बात कर रहे हैं केवल कि इनिशियली किस तरह का स्टेन लिया, इंटरमीडिएट में कौन सा लिया, लेट में कैसा हो जाएगा. क्लियर? इनिशियली इट वाज बेसोफिलिक, in between it is polychromatophilic. Later on, maximum of the cell cytoplasm will have only hemoglobin. Okay, so maximum of the acidic stain will be taken. Okay, now the cell will become orthochromatic erythroblast. It will take only the eosin stain and will get converted into orthochromatic erythroblast or the नॉर्मोब्लास्ट. इसकी टर्मिनोलॉजीज को ध्यान में रखना और किस स्टेज पे क्या चीज आ रहा है. ठीक है, देन आफ्टर दिस डे 4 इज द स्टेज वेयर द नॉर्मोब्लास्ट और द ऑर्थोक्रोमैटिक एरिथ्रोब्लास्ट हैज बीन फॉर्म्ड. ओके, नाउ दिस इज टेकिंग प्लेस इन द बोन मैरो. इट हैज टू एंटर इनटू द सर्कुलेशन. Okay, now when it has to enter into the circulation, the capillaries, they are of broadly 3 types. They have fenestration, they have sinusoids, and they are in continuation. Okay, so these capillaries we are residing near the bone marrow is of sinusoid nature. So through diapetesis, इसका क्या है, biconcave nature हो जाता है, तो क्या squeeze हो जाएगा easily. So it will squeeze or through diapetesis it will reach into the capillaries and enter into the circulation. But when it is entering into the circulation, at what stage it is entering? It is entering at an immediate immature stage of erythrocyte, that is known as reticulocyte. किस स्टेज पे सर्कुलेशन में एंटर हो रहा है? रेटिकुलोसाइट स्टेज पे. और रेटिकुलोसाइट क्या है? इट इज एन इमीडिएट इमेच्योर स्टेज ऑफ एरिथ्रोसाइट. ओके, and then within 1 to 2 days this reticulocyte will get converted into a mature RBC or erythrocyte. Simple story, but now we'll make it a complicated a bit. Okay, हमने अभी केवल क्या पढ़ा था, इसका केमिकल नेचर जो कि चेंज हो रहा था by doing the various stains. Okay, गेट खोल दो. They want to come, open the gate. Okay, so initially it was basophilic in nature, then it turned out to be polychromatophilic, later orthochromatophylic. Clear? Then comes the reticulocyte and then comes the mature RBC. Clear? This was the chemical change. Another important change which you can observe is smaller size. Okay, decrease in the cell size. Another important change, nucleus. Okay, finally at the stage of normoblast it becomes a condensed form of nucleus, and that has to be excluded or removed from the cell. Okay, pycnotic nucleus, and that has to be removed from the cell. If it is not removed, then it will not result into reticulocyte or a mature RBC. Clear? Okay. Now for this process to take place, okay, stem cell, committed stem cell, phase 1 ribosome synthesis, second is hemoglobin accumulation, and third is ejection of nucleus, reticulocyte, and this is the RBC. This diagram is what has been asked or what has been expected from you when you are answering your question related to erythropoiesis. Clear? If you have color pencils, you can prominently draw and show. If you don't have any color pencils, at least label it properly which part is nucleus, which is ribosomes, and where the hemoglobin synthesis is getting started and how. Now this ये दिख रहा है ना ब्लू कलर का, ठीक है, ये रेटिकुलम है। अपन ने इसको रेटिकुलर साइट क्यों बोला है, क्योंकि इसके अंदर रेटिकुलम दिखता है अपने को। विल सी इन लेबोरेटरी रेटिकुलर साइट काउंट अपन करेंगे और लेबोरेटरी में देखेंगे, बट दिस इज द थिंग विच इज सीन रेटिकुलर साइट. This has been seen with a special stain technique. This we do in our practical class as a total reticulocyte count. Now, what is the normal total reticulocyte count? Picture मैं वैसे दे दूंगी, ठीक है ना, ये भी मतलब पिक्चर वैसे दे दूंगी आपको, ठीक है। चलिए नेक्स्ट, नॉर्मली ओनली 1% ऑफ द आरबीसी या ब्लड टेस्ट कराते हैं तो अपना जो आता है रेटिकुलर साइट काउंट प्रेजेंट इज 1%. Okay, if there is more than 1% of the reticulocyte present in the blood count, okay, what term will be used? Normally only 1% of the reticulocyte is present in the normal blood count. If this count increases of reticulocyte, okay, what term is used? Today your orientation is not there in the class, that's why. If there is any increase in the any kind of cell, what term is used? Cytosis. Reticulocytosis. ठीक है ना, तो उसको अप्लाई करना आना चाहिए हमें, है ना, आज ओरिएंटेशन नहीं है, बैठो. So that is known as reticulocytosis. If there is an increase in the reticulocyte count, that means there is increase in the rate of erythropoiesis. Now that increase in the rate of erythropoiesis may be due to anemia, okay, may be due to going to high altitude, okay. There are a number of reasons which might result into hypoxia that will be a triggering factor for erythropoiesis. But increase in reticulocyte count, they shows that there is increase in the rate of erythrocyte formation. Normal count of reticulocyte is, just remember, it is 1%. ठीक है, और इससे ज्यादा हो रहा है, that means there is increase in the rate of formation of RBCs or increased rate of erythropoiesis. Okay. Now, how this whole process is regulated? So the question comes: define erythropoiesis, write down the various stages of erythropoiesis, what are the factors which affect erythropoiesis, or add a note on regulating factors. Okay, so when we talk of this, okay, regulation, general factors, okay, we have divided the factors into three categories. First is the general factor, second is the special maturation factor, and third is the hormonal factors. When we are talking of general factor, most potent stimuli responsible for erythropoiesis is hypoxia. Okay, some of the students might be liking to go for trekking, okay, है ना, और अपन हिली एरियाज पे जाएंगे, हाई एल्टीट्यूड पे जाएंगे, तो डेफिनेटली देयर विल बी लो ऑक्सीजन ओवर देयर. When there is low oxygen concentration, definitely the requirement or the hypoxic condition will result. This hypoxia will be acting as a stimulating factor for erythropoiesis. Another condition may be anemia, decrease in the RBC count, decrease in the hemoglobin concentration. Okay, so when there is anemia, then there will be hypoxic condition that will act as a stimulating factor. So the most potent stimulating factor for erythropoiesis comes out to be hypoxia, and especially renal hypoxia. Okay, that we'll see in detail. So hypoxia is the most potent stimulating factor for erythropoiesis. Okay. Another factor which we'll talk about is special maturation factor. So when we are seeing the different stages of erythropoiesis, okay, सबसे इम्पोर्टेंट कंटेंट क्या बनाना है इसको, हीमोग्लोबिन, ठीक है, और बाकी सारे सेल ऑर्गेनेल्स जो प्रेजेंट है उनको बाहर करना है, खत्म करना है, न्यूक्लियस हटाना है. तो मेन कंटेंट क्या है इसका, जो कि हीमोग्लोबिन को सिंथेसाइज करेगा. हीमोग्लोबिन सिंथेसाइज के लिए अपने को सबसे इम्पोर्टेंट क्या चाहिए, वी रिक्वायर आयरन, वी रिक्वायर प्रोटीन, वी रिक्वायर कार्बोहाइड्रेट्स, वी रिक्वायर फैट्स. ओके, with the combination of succinyl coenzyme, pyruvate, and we result into the formation of 9-photoporphyrin plus combination of iron results into the formation of hemoglobin. So we require everything for formation of hemoglobin, but they act in a very smart way. Okay, if iron is present in the free form or it is not bounded to any protein in the body, okay, then what will happen, it will result into the formation of free radicals via Penton's reaction. So whenever they are present in the body, it has to be present or it has to be combined with some or the other protein. So that pathway अब तो बहुत कम हो गया आपका syllabus, so but that pathway you will study in biochemistry. So please try to remember that pathway of iron absorption, how the iron is being absorbed or taken by the body and goes to the different area, whether for utilization or for storage. Okay, so I'll just try to cover the physiological aspect, but you keep in your mind, because if you are not clear with the biochemistry part, it will be difficult for you to understand the mechanism. Okay, until unless we reach to the molecular level, it is very difficult. आप उसको केवल रटते रहोगे चीजों को, you will not be able to understand the thing. When we are talking of special maturation factors, the dietary factors, Cassell's intrinsic factor and the extrinsic factor, hormonal factor, thyroxine, testosterone and growth hormone. Now when we are talking of general factor, hypoxia, okay, so what will the hypoxia does is, I have told you that mainly it is the renal hypoxia. So what will renal hypoxia do? Okay, when there is hypoxia going to the renal circulation, okay, renal tissues with renal hypoxia ho gaya, then what will it do? It will be resulting into the secretion of erythropoietin. Okay, it is a glycoprotein, it is a hormone, so it is being secreted from the kidneys. Now the exact original cells from where it has been secreted is still a mystery, but different theories have been given for this. Some theories suggest that it has been secreted from the peritubular capillaries. Some theories suggest that it has been secreted to the endothelial cells of the renal epithelium. Okay, so depending on this, 90% of the erythropoietin comes from the renal or the kidneys. 10% of the erythropoietin comes from liver. So if a person is having some sort of renal problem, acute, okay, or chronic, resulting into renal failure, then there will be problem in the erythropoiesis. 90% of the erythropoietin is coming from the kidneys, only 10% from the liver. So this 10% will result into the 30 to 35% of the erythropoiesis. When that much is the erythropoiesis, what will happen to the hematocrit value? It will be less. The hematocrit 40 to 45 था, वो हो जाएगा 20 to 23%. ठीक है, सबसे इम्पोर्टेंट स्टिमुलेटिंग फैक्टर हो गया हाइपोक्सिया. ओके, हाइपोक्सिया क्या करेगा? इट विल आस्क द किडनीज टू सिक्रेट erythropoietin. Okay, erythropoietin will stimulate for the formation of erythropoiesis. Okay, now this erythropoietin, at what point it will act? It will act at the committed stem cells which have been committed for erythropoiesis. Okay, so the committed stem cells E, okay, that is for erythroid series, that will be responsible for mitotic division of these cells. Okay, they are responsible for the mitotic division of these cells or series, which will be resulting into mature RBCs. For any type of cells to divide mitotically, okay, the nucleus has to divide, it has to require DNA, it has to require the nucleic acid content, okay, and other factors responsible for the division. Okay, for the formation of DNA, special thing which is required is vitamin B12 and folic acid. Clear? Vitamin B12 and folic acid. हम इसको क्या ऐसे separate कर नहीं सकते, but just for our study sake we have divided it into 3 major categories that will be regulating the erythropoiesis. Okay, but when we are talking we have to just correlate it. Okay, that the dietary supplement come into action, what is the role of that supplement. Okay, so when we are talking of this B12 and folic acid, they are required for the nucleus, okay, when the mitotic division is taking place continuously. Okay, so if there is problem in this vitamin B12 or folic acid, what will happen? There will be no proper maturation or growth of erythroid series. So the cell will remain bigger in size. Okay, when the cell is bigger in size, what we call it? Macrocytic or megaloblastic. Okay, so the anemia occurring due to deficiency of B12 or folic acid will have which type of cells? When we see it, it is megalocytic or macrocytic. है ना? तो आप उसको कोरिलेट करोगे तो इसको समझना इजी है कि अपन इसको कैसे कर रहे हैं, कहाँ से सबसे पहला इफेक्ट आ रहा है. एरिथ्रोपोइटिन भी सर्कुलेशन में तब आता है जब उसकी एरिथ्रोपोइटिन इफेक्टिव एक जीन होता है, उसको हाइपोक्सिया स्टिमुलेट करता है, ट्रांसक्रिप्शन करता है, ट्रांसक्रिप्शन से एरिथ्रोपोइटिन का सिक्रीशन होगा और फिर हाइपोक्सिया का करेक्शन होगा. दिस मॉलिक्यूलर लेवल इज नॉट बीन गिवन इन योर बुक्स, और होगा तो 1-2 लाइन होगा, but we are not that much concerned in detail about this. But how this erythropoietin is governing, that has to be clear, from which source it is coming, and if there is deficiency or decrease in their secretion, what will happen to the erythropoiesis. Clear? ठीक है, तो सबसे important factor अपना erythropoietin, its source, its effect on which particular type of cells? Committed stem cell that will affect into the pro-erythroblast. So what it is going to affect? It is going to affect the division of the nucleus and the cell. Okay. Special maturation factor, dietary factors में क्या क्या आ गया? Vitamins, proteins, essential amino acids. Okay. Which important vitamins are required? Vitamin B12 and or vitamin C is also required. Okay, vitamin C, vitamin B6, okay, B12 and folic acid. Okay, इसका mechanism पता है B12 का? The mechanism, how it is being taken by the body, how it is being getting absorbed. The requirement of the vitamin B12 is in micro quantity, okay, and its daily intake is in milligrams. Okay, so the deficiency of B12 takes 3 to 4 years to appear. Okay, it has a capacity to store 1000 times what is required, so its deficiency takes years to appear. Okay, if there is deficiency, that has to be appeared. Okay, when we are talking of different cells, they have different lifespan. So in case of these different blood cells, the platelets have the smallest lifespan. So if there is deficiency of this type of vitamin, okay, the first symptom that will appear will be purpura. Okay, because which cell is having lifespan shorter? The platelet, which is very rare, the vitamin B6 deficiency anemia, क्योंकि इसका स्टोरेज बहुत अच्छी क्वांटिटी में होता है और रिक्वायरमेंट इज इन माइक्रो क्वांटिटी. Okay, but if there is deficiency, the first symptom which will appear will be of purpura. Okay, and how it has been taken? It has been taken through the diet. Okay, it enters into the GI tract. Okay, the parietal cells of the stomach. Okay, they are responsible for secreting the intrinsic factor. B12 will combine with the intrinsic factor. If it does not combine with the intrinsic factor, what will happen is the pancreatic secretion which is coming into the small intestine will digest it. So this will combine with the intrinsic factor, reaches to the ileum where there is a special protein or the receptor which will attach with this of intrinsic factor B12 combination, and this will change the transformation and the vitamin will be taken inside and the intrinsic factor will have to be eliminated or removed from the body. ये इतनी खांसी आती है या आप लोग करते हो? आती है तो तुम लोग ट्रीटमेंट नहीं ले रहे क्या? ठीक है, तो इसका जो मैकेनिज्म है ना, ओके, विटामिन का ये है. और फॉलिक एसिड, इट इज आल्सो रिस्पांसिबल फॉर द फॉर्मेशन ऑफ न्यूक्लिक acid or DNA. So if there is deficiency of this folic acid, it will result into a bigger nucleus and a bigger cell size. Okay, so it will also result into megaloblastic type of anemia. So to make the picture clear, the serum level of folic acid or the vitamin B12 has to be done separately to make it very much specific that which type of anemia is present. Because the picture, when we see the peripheral blood film, it is the similar picture, megaloblastic, but when we have to clear it out which type, the serum level has to be estimated differently. Okay. Then the hormones other than the erythropoietin is thyroxine, testosterone and growth hormones. In case of testosterone, this is one of the reasons why the RBC count is more in males compared to females. It has a stimulatory effect of erythropoietin. Okay, hypoxia, most potent stimulus, causes release of erythropoietin. Most dependent stage is colony forming unit erythroid series to pro-normoblast. Okay, increases the vitality of erythroid precursors by preventing DNA cleavage. That is the normal blood oxygen level. If the balance gets disturbed, starts the stimulus hypoxia due to decreased RBC count, decreased availability of oxygen to the blood or increased tissue demand for oxygen. Either may be the reason for hypoxia. Okay, from the kidneys, the erythropoietin is getting secreted, stimulates the red bone marrow and increases the count of RBC. Okay, and correction is done by increasing the oxygen carrying ability by the blood. This is the simple flow chart showing the erythropoietin mechanism. In physiology, you cannot write the whole chapter when you are coming to write answers. The most simple and effective method of writing an answer is either go for a diagram or a flow chart or tabulate it. Okay, if this is the thing, if there is decrease, it will result into this. If there is increase, it will result into this. So by this, we are able to know that your concepts are clear, because you cannot write the whole chapter in your copies, but the concept has to be there, the content has to be correct. Okay. Special maturation factor, dietary, proteins, iron, magnesium, copper, okay, cobalt, nickel, these are required for early normoblasts. Okay. Vitamin C, folic acid, Cassell's intrinsic factor for maturation, released to the oxyntic cell of stomach. Now another MCQ question, from where the factor is being released is from the oxyntic cell of stomach. Extrinsic factor, vitamin B12. Okay. Now in the next class, we'll talk about the fate of RBCs, or what happens to RBC after completing 120 days. Okay. कितना टाइम हो गया? बस चलो, तो आज ही खत्म कर दें क्या? क्यों, क्या हो गया? क्या? जब कुछ पूछो तो इतने इनएक्टिव हो जाते हो। तुम तो आज सो रहे थे क्लास में, ठीक है? You were not oriented that much. Okay. So in our practical class, by next week we'll start. Write on your name, your roll number, and your sign. Okay. In your next class, we'll start with the counts. Okay. We have done hemoglobin. Next we'll start with counts. We'll do RBC count, WBC count, and platelet count. So in this week, we'll try to cover the theory aspect of WBCs and their counts. Okay. So you should be clear. For that counting, we require a special type of instrument, that is Newbot chamber, that will be required in your laboratory. Okay. With the help of this Newbot chamber, we'll do the different counts. Different counts means RBC, WBC, platelets, okay, eosinophil, okay, separate different counts. Either they will be in demonstrative exercises or they will be in practical. Okay, so RBC, WBC, that comes in your exam, either RBC count or WBC count. So in the similar way which you have done the hemoglobin, you have to prick yourself and do the experiment. If you remain absent, it will be assumed you have done it. Okay, so no repeat class. So don't try to remain absent. चला गया, उधर चली गई, class हो गई. Functions of RBC you can write or elaborate more. Okay. Thank you.